All right, ladies and gentlemen, welcome back. So we're going to continue talking about biotechnology by picking up with DNA fingerprinting today. So remember when we talked about the Human Genome Project, we said when we look at individuals in the human population, we know that 99.9% of your DNA is identical to everyone else. And those presumably 99.9% of your genetic makeup is information that requires you to be human. So evolutionarily, it makes sense. If I need that information to allow me to function as a human species, then that information probably is not something that I want to have a lot of variation in or want to change a lot. So, that being said, there's still that 1/10 of a percent that does make you different from everyone else. And that 1/10 of a percent is what DNA fingerprinting takes advantage of. So, DNA fingerprinting will analyze sections of your DNA that we presumably assume have little or no function to them. So, remember, we talked about those chunks of DNA that seem to be non coding, I mean, they don't create proteins. So, when we go back to evolution, remember that protein really is the same thing as saying phenotype. So, all of your genomic information, your genotype, what's coded for in your DNA, then corresponds to the physical characteristics that you have. Those are the proteins, that's your phenotype. And when we talk about evolution, evolution works on those phenotypes directly. It's going to determine, all right, you're really good at camouflaging yourself, so you stay away from your predators. So that set of proteins, thus the DNA that codes for it, should be more advantageous and as a result should stay around in the population. But if you have sets of DNA that don't code for proteins, as a result, don't contribute directly to your phenotype, your physical characteristics, then evolution doesn't know it's there. Natural selection can't work on it. And so that 1/10 of a percent is more than likely the pieces that don't code for proteins as a result have never been worked on from a natural selection standpoint, and as a result have a lot of variation within them. That's what DNA fingerprinting is going after, and those pieces of information, those pieces of our genome that we know vary widely from one person to the next, are these genetic markers that we're going to utilize for DNA fingerprinting. Quick caveat I'll put here, when we talk about that term genetic marker, it can get used in a couple of different scenarios in a couple of different ways. We're going to talk about it here today as a way to talk about sections of DNA that have a lot of variation within them. There's a lot of differences, but you may also come across that term genetic marker as sort of a mutated version of a normal copy of a gene. So I may have mentioned that term before when we talked about genetic testing and we're looking for a genetic marker for breast cancer, for example. And that genetic marker may or may not be as widely varying from one individual to the next as we're talking about here, because those genetic markers are specifically mutated sequences that we know are associated with certain diseases. So hopefully that makes sense. Today, we're going to use that term genetic marker just to mean a section of DNA that's got a lot of variability in it because it doesn't actually code for any protein. So, DNA fingerprinting has actually been a piece of technology that we've been using, at least here in the US, since the 1980s. Big popular piece of evidence that came up, especially when we talked about criminal cases, paternity cases, that kind of thing. And the technology has only gotten better and more accurate since then. So, when we talk specifically about the technology underlying this concept of DNA fingerprinting, we've got to talk a little bit about PCR or polymerase chain reactions today, as well as this thing called gel electrophoresis. So let's start with PCR. So PCR, polymerase chain reaction. Hopefully that term polymerase looks familiar. Remember, it ends in an ASE, so we're talking about an enzyme here. And we talked about polymerases when we talked about DNA replication and when we talked about transcription. You had your DNA polymerase in DNA replication and your RNA polymerase in transcription. Well, today we're specifically going to talk back about that DNA polymerase. And remember, DNA polymerase's job was to take DNA in your cell. That cell is getting ready to go through cell division, so we have to make another copy of DNA in order for that new cell to have the set of instructions that it needs to function. So essentially what polymerase chain reactions are, or PCR is taking that DNA replication process out of your cells and doing it in a test tube on a lab bench. So it's really kind of a neat idea. So when we look at these machines, and this is kind of an old one, we actually have one here at school that maybe is more like this. It kind of looks like maybe about the size of a typical coffee maker, like old school standard coffee maker, kind of laid on its side. So it's not particularly huge. But what it's able to do is to take even a really small amount of DNA. We're talking less than a microgram. So 1 millionth of a gram is necessary at minimum in order to do this. But then I can take that DNA sample and make billions and billions of identical copies of it, really within a couple of hours. So that's pretty crazy. I can also be very specific about targeting just the sequence that I want with the help of restriction enzymes, and we'll talk about these things called primers in just a second. So this is the basic process of how PCR works. So I take a test tube, and in fact, it's a really tiny little test tube. If you kind of look at your finger and look at maybe about the width of your fingernail, give or take, that's how tiny this test tube is. Particularly, say your thumb, and I've got small hands, so all right, it's a pretty tiny test tube. But what we do is we take that test tube and we've got to fill it with certain things. So remember, I'm doing DNA replication outside of a living cell, which means I need to have in that test tube the materials that are necessary to go ahead and build more DNA, just like your cell would have. So I'm going to go ahead and put in that test tube the target DNA that I have, so the DNA sample that I have. I've got to put in a bunch of free nucleotides, so A's, T's, C's, and G's, because if I'm going to build more DNA, I need to have building blocks to do it. I'll also put primers in there, and those primers are very specific sequences that I know will end up complementary to portions of DNA in this target sequence. And then I also need DNA polymerase. I need the enzyme. So all of that goes into a test tube along with some buffer, and then we stick that test tube into a machine. And that machine then, its job is to be what we call a thermocycler. So break that term down. Therm means heat and cycle is literally to cycle. So the idea of that machine, really, in order to allow this to happen, is to turn up and down the temperatures at specific times and at specific variations to allow this DNA replication process to work outside of a cell. So what does it do? Well, the first thing it's going to do is that I'm putting in double-stranded DNA. So I need to be able to denature it. I need to be able to allow that double-stranded DNA to separate. So I'm going to heat that DNA up really hot. In fact, it's usually about 95 degrees Celsius. Now, take notice, I said Celsius, not Fahrenheit. So your body temperature is roughly 97, 98 degrees Fahrenheit. But when we talk about Celsius, really quick, easy, quick conversion, not perfect, but it does a nice job, is to take that number, double it, and add 32. That gets you a pretty close estimation of what we're talking about in Fahrenheit. So if I'm talking 95 degrees Celsius, double it. So now I'm talking, what, 180, so 190 plus 32. So let's see, that takes me to 200, 210, 220, about 295. Scratch that. 225 in Fahrenheit. So it's hot. All right. But what that's going to do is force that DNA to separate. Once that DNA is separated, we allow that whole mixture to cool back off. All right, and that slight cooling, it takes it down to about 52 degrees Celsius, give or take. What that's going to do is allow those primers to now stick, or what we call anneal. They're going to go into that single-stranded DNA and now find the complementary base sequence hydrogen bond. And once that happens, once those primers stick, this DNA can't come back together again. which is the idea. We'll then turn the temperature back up a little bit and allow that DNA polymerase to now function at its maximum potential. Remember that when we talked about enzymes earlier this year, that enzymes work at a very specific range to be most efficient. So we're going to bring that thermocycler back up to the temperature that allows that DNA polymerase to be most effective. Side note here, remember I just said I've got to heat this up to 95 degrees Celsius. So we're talking almost double, if not more, human body temperature. That means that DNA polymerase that we're putting in here is not from a human cell. In fact, it's from DNA that is found in, or excuse me, it's from a species of bacteria that is found in the hot springs in Yellowstone. That's where it was originally discovered. Now we culture it in a lab and just extract the enzyme as we need it, but that's actually where it was originally found. So it's actually in what we call a thermophile, a extreme bacteria that really likes the heat. So that enzyme is then stable at those high temperatures. Because if we tried to put in a human DNA polymerase, well, we go heating it up to 95 degrees Celsius, and that DNA polymerase would just denature. It would fall apart right along with the DNA, and that's not the effect we're wanting here. So just kind of a side note. So we put all this material into this test tube, heat it up, denature the DNA, cool it back down to allow those primers to anneal or to bond. Then we warm it back up again and allow the DNA polymerase to do its job by adding those free nucleotides and complementary basing, building the strand. So presumably, if I put a single DNA molecule into this, after I finish that first heat, cool, heat cycle, I've now got 2 copies. The thermocycler is now going to recycle this set of steps again, but now I'm starting off with 2 copies. So we heat it up, denature those 2 strands, cool it back down, allow the primers to attach to those 2 strands, heat it back up to allow the DNA polymerase to make copies of it, and now we're up to 4 strands. And we do it again, and then we're up to 8. And we do it again, then we'd be up to 16. Do it again, up to 32, so on and so forth. And this will do this for potentially hundreds of cycles until we get enough DNA to actually be.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So once we've got a sample that we can work with, then we can go ahead and actually do some analysis on it. And that analysis is going to come in the form of this technique called gel electrophoresis. So we'll take those DNA sequences that we just ran through that PCR and we'll hit it with a restriction enzyme. We'll chop it into pieces and we'll then run that, what we call digested DNA, through a gel electrophoresis. And the point here is that this gel electrophoresis technique allows us to sort molecules by fragments and sort them specifically by length. So how long are these DNA molecules, these DNA fragments, based on the number of base pairs that are there? So, like I said, we'll initially cut that sample with those restriction enzymes. Remember, those enzymes are going to cut at specific sequences. So let's assume we're working on a mugging case, right? So person gets mugged, they happen to get their attacker, um, scratch them, so they've got some of their attacker's cells under their fingernails. So the forensic scientists will go in, they will extract those cells from that victim's fingernails, they will isolate the DNA, they'll go through PCR to make lots and lots of copies of those sequences that they know tend to vary a lot between people. And then they'll hit that with a restriction enzyme. They'll do that same process of extraction, PCR, and then restriction digesting with their suspects. So let's just say we've got 2 potential suspects. So now I've got 3 samples that I'm working with. The one that I extracted from the victim and the 2 DNA samples that I've taken from my 2 potential suspects. I'm going to do the same restriction enzyme on all 3 samples so that they theoretically should all cut in the same place, assuming that that DNA sample from the victim matches one of those suspects. Once I do that, then I'm going to load those DNA samples into this gel electrophoresis chamber. So this really just is a big plastic chamber. Okay, so you're sort of looking at it as a cross section. And sitting in an elevated piece in the center is where we put this gel. And it kind of is like the consistency of Jello. But when you zoom into it molecularly, it looks like a big matrix or a big maze. So that DNA has to weave its way through all of these sort of roadblocks that are in place until they can make it to the other side. So those samples, we create little divots, little wells, indentations into the gel. We'll load those samples in here, and then we attach this to electrodes. So it produces an electric charge. And that's really important because that electric charge is where that electrophoresis piece comes from. And though that electric charge is what's going to force these molecules to move through this gel in a very specific orientation. So here's sort of a top-down view now, right? So here are these divots, here are these wells, these indentations, and we're going to load our DNA samples in here. So victim and my 2 suspects, if we stick to that mugging case example. And because this is a big matrix, a big maze, the DNA has to weave its way through here. Now, DNA happens to have a slight negative charge overall to the molecule. If you remember the way that DNA molecule looks, and you go back and look at those phosphate groups, they've got negative charges on them. So we're going to load our DNA samples on the same side as that negative charge that we're putting in here. So, right, if you go back here, I'm going to put that negative electrode here, positive electrode here. And that's exactly what you're seeing here. So here's the negative charge, positive charge. And if I put something negative here, right, like charges, just like everything, they will repel, while unlike charges, opposite charges will attract each other. So by ensuring we put the DNA in this end, because it's negatively charged, it's going to then get attracted to the positive and it will run through that maze, that matrix here in the gel, and separate out those samples based on how big they are. While I'm on the idea of those charges, if I were to put some other molecule in there, say maybe a protein or some sort of dye or anything else, if it's not negatively charged, then I have to be aware of which direction I had put those wells. So if I were to put a protein in here that had an overall positive charge, then I wouldn't want to put it here because what's going to happen is it's going to push it this direction and you're going to end up pushing it out into that solution, buffer solution, basically water and kind of soap, back this direction, and I'd never be able to see any result. I'd have to flip it around and put the wells down here so that it ran from positive to negative instead. So just something to think about when we're talking about what kind of samples can go in here and making sure that we're running it appropriately. Anyway, we're going to talk DNA. So DNA is negatively charged, so the wells go towards the negative end, and we push them towards the positive. So remember I said this is kind of like a big maze or big matrix. So another way to think about this is that it separates it out by size because there's resistance, right? So maybe not the most politically correct analogy, but kids seem to have it make sense to them, is sort of think of this like a swimming pool full of jello, right? And I put a really skinny kid in that pool and said, get to the other side. At the same time, I put a heavier, chunkier, rounder um, guy into the pool and said, you have to make it to the other side as well. Well, who's naturally going to be able to get through that jello and get to the other side faster? All right. More than likely, the skinny guy, because he's just going to hit less resistance, there's less friction. And that's kind of the same idea here that DNA has less resistance getting through these small cracks and crevices, making its way through the gel than this bigger, longer, chunkier DNA does. So as a result, those gels, as they get run for a certain period of time, the smaller DNA hits less resistance and can get through the DNA gel faster, can get through that gel matrix faster. And so they will get further away from the well that they started at. So here's the well where we started putting the sample in, all right? In the same amount of time, let's say it takes an hour, this DNA, because it's so big, could only get here, but the smaller piece could make it further down the gel. Something else to think about when we talk about gel electrophoresis, depending on the way that, or the molecules, I should say, that you're putting through here, a lot of this stuff is colorless. There's no way to see it. So DNA in particular, among other things, needs to get stained. So we will put, once this gel is run for, say, an hour, we'll then put it into a liquid stain solution and allow it to stain so that that stain specifically is designed to stick to that DNA so that we can see it. Either it's usually either some sort of blue stain and then I can just see it visually, or it is something like something called ethidium bromide, in which case that is activated by UV light and it glows. So it just depends, but usually you can't take this gel right out of the apparatus and be able to see it if we're talking something like DNA. And so we have to stain it afterwards. So again, the result here is that the larger samples are going to run slower, stay closer to the well, the smaller stuff can travel more quickly and thus get further. So if I go back to that mugging case, what I'm going to do is take a look at these pictures, right? These are pictures of gels, so here's those original wells that we would have loaded those samples in, and I allow the things to run. So if I cut them with the same restriction enzyme and they came from the same person, then those gels and those lines should match. So if I look, all right, now keep in mind this is slightly tilted, right? So it's not a perfect image. But it looks like this and this are probably about the same distance, but here to here they don't match. So probably not from the same person. But if I take a look at maybe this one to this one and this one to this one, now maybe they do match, and now maybe I start to consider that coming from the same individual. And that's the idea. We're looking for these banding patterns to match up if they came from the same person. So if I had suspect 1 and the DNA from my victim under the victim's fingernails, if they have the same banding patterns, and I know that that DNA came from the same person. So here's what we're talking about then. So we use those DNA fingerprints, right? Here's just more of a diagram to make it a little easier to understand what I was saying. So here's that crime scene sample, right? Presumably, say, the DNA taken from under that person's fingernails. I've got 3 suspects in this one. And now if you look, these banding patterns match. Why do they match? Because they came from the same person. So when I hit them with restriction enzymes, the enzymes cut in the exact same places because the DNA sequence is identical. So that when I ran them through that gel electrophoresis, those bands, because they're the same length of base pairs in each of those bands, each of those segments that got chopped up, they are in the same location and thus they match. And we can use this for crimes. We can also use this for paternity tests. So if somebody's questioning whether or not that individual is their father, we can run a paternity test. Now, there's an interesting one because that paternity test, the bands won't be identical. because you're talking child who has two parents, mom and dad, who both contributed their DNA to that person, to that individual. So it won't be a completely identical match like you see here in the picture with the crime scene samples. Instead, you'll get some of the bands that match mom, and then the rest of them will match dad. And so you have to piece it together a little bit more. But it's usually pretty obvious, at least in sort of classic textbook examples, to figure out who's the father. The other thing is that the DNA can really come from any number of sources. So it can be taken from your blood, so long as you've got white blood cells, by the way. Your red blood cells actually don't contain any DNA. But we can take it from semen, we can take it from hair, so long as you have the hair follicle, the actual piece that attaches into your scalp. The actual hair itself, if I were to just come over and cut a piece of hair off your head, doesn't actually contain any functional DNA anymore. But your bone, skin cells, all sorts of things.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about all of molecular biology. Before I get there, I want to talk about a story close to home. In the 1970s, some researchers went to Yellowstone Park. They were in the geyser basin. They pulled these bacteria out called Thermus aquaticus, and from that, they pulled out an enzyme called Taq polymerase. That means Thermus aquaticus polymerase, or the DNA polymerase found in this bacteria. Cool thing about it is that it can handle really high temperatures and survive. And the whole genetic engineering molecular biology is built upon this enzyme. So we're talking about trillions of dollars. How much of that goes back into Yellowstone Park? None. And so they've changed their policy on that. But it's a great way to kind of frame this idea of molecular biology and how important it is. When I was thinking of how I wanted to explain this in a way that made sense, I always kind of fall back on an analogy. And so basically what I want to talk about is how we've gone over the last 30 or 40 years to the point where we now know all the DNA inside a human. And so basically, think about it this way. Imagine if you look at these magazines right here, there's a whole bunch of information that's found inside here. There's a whole bunch of text inside here. And basically, if I could take all of the text that I want and make something out of it, like a really cool ransom note, then I would be manipulating that text. And so I want to kind of use this as an analogy for what we've done in molecular biology. And so what was the first thing that was ever done? Well, the first ransom note ever in the terms of molecular biology looked something like this. And you might say, that's not very impressive. Basically, what is it? It's like cutting out letters from a magazine, but turning it upside down so you can't see the letters. And so what was the first thing that we were able to do is we were able to cut the DNA. And we do that using a restriction enzyme. But you should think of a restriction enzyme like a scissors. It's able to cut the DNA. How do we paste DNA? Well, basically to paste DNA, the glue stick equivalent of that is hydrogen bond. DNA will just come back together again once it's been cut because of the hydrogen bonds. What's the next ransom note? Well, it would look something like this. Basically, it was the first recombinant DNA. We took DNA from a frog, if I remember right, and a bacteria, and they were able to combine those two using this hydrogen bonding. But again, a not very impressive ransom note. Next thing that happened was something like this. So you can see in here that what we're doing is we're actually ordering these little fragments cut out of a magazine from small to large. But again, you can't see what the letters are, so that's called gel electrophoresis. And then we developed something called PCR, polymerase chain reaction, where we made a copy of that. And so if this is that original, in PCR we make a duplicate of that. And we make an exact duplicate. So every duplicate that we make from that is going to be exactly the same. And that's what PCR does. It makes copies of DNA that's exactly the same. Okay, what's the next advancement? Well, the advent of the marker. So a marker is basically going to be a little section of DNA that marks that specific DNA. So if you read that they've found the marker or the genetic marker for breast cancer, that means that there's a piece of DNA that's inherited by all people who have this genetically based breast cancer. And then finally we get to where we are today. So right now we've used DNA sequencers to figure out all the letters inside our DNA, but it's hieroglyphics. In other words, you can't really read what it means. We know what all the letters are, but we don't know where the genes or what the genes express. In other words, what proteins they make. And so when we look at this ransom note, this is the future. This is where we want to get, but we're still quite a ways out. And so basically going back to the big things in this talk that you'll need to remember. First one is the scissors. The role of the scissors will be played by restriction enzymes. The role of the glue will be hydrogen bonds that are found within the DNA. The ruler or the measuring device is something called gel electrophoresis. To copy that, we use the polymerase chain reaction. And then to actually read it, we use something called a DNA sequencer or a gene sequencer. It's going to look at all of the letters. Even though we might not know what they do, we can find them right now. So let's start with the scissors, and that's a restriction enzyme. Where do restriction enzymes come from? Well, basically they come from bacteria, because bacteria have been locked in this million-year war with viruses. And the viruses, you can see, inject their DNA into the bacteria DNA and they make more viruses. And so how do you fight back? Well, basically what they do is they methylate their DNA. First of all, what does methylate mean? You basically add a methyl group to all of their DNA from the bacteria to protect it, and then they're going to secrete these or create these restriction enzymes. And what the enzymes do is they chop up or cut DNA. Since all of the bacterial DNA is protected by these methyl groups, what it's really going to chop up is all this foreign DNA. And so all of that viral DNA is broken apart, and then the bacteria, after it's done that, can return to its specific shape. So let me show you how a restriction enzyme works. So if this is a section of DNA, a real common restriction enzyme is something called EcoR1. It comes from the word E. coli. This is a restriction enzyme 1. And basically what it'll do is it'll scan the DNA until it finds this specific sequence, and it's literally going to cut it in half. And so if I were to find that, where is that going to be up here? Well, you can see we have a GAAT, so it's going to cut right like that through here. It's going to go all the way to the end, and it's going to cut it in half like that. And so if we take a look at that, basically what will it do? It will break that into 2 fragments. If we take another one, oh, well, did you see that? It came right back together again. So what brings it back together again? There are going to be little hydrogen bonds here between those 2 ends. And in fact, we call this a sticky end and this a sticky end because there are going to be hydrogen bonds formed between the 2. And so once the enzyme gone, it comes right back together again. If we look at another one, this is the HindD3 enzyme. Basically, it's going to cut between these 2 A's, and so it would cut it right here, all the way down here, and it's going to cut it into 2 fragments like that. If we apply that enzyme that was found in this specific type of bacteria, if it goes away, then it comes right back together again. What if we add both of those enzymes? What's it going to do? Well, it's going to cut it into 2 points or 2 restriction sites, and then we're going to have 3 fragments that come from that. So those are restriction enzymes. What do we use them for in molecular biology? Basically to cut DNA, and then to glue it back together again, all you need is hydrogen bonds. And so the first recombinant DNA, that of a frog and a bacteria, how did they get together? Basically, they cut them both with the same enzyme. They had the same sticky ends, and then they were able to come back together again. What's the next one that we have to do? So what we've done is shown you how to cut, that's a restriction enzyme, how to glue, that's hydrogen bond. Now we have to figure out how to separate the DNA according to its length. And so to do that, let me talk about pachinko machines. Pachinko machines are essentially these vertical machines. They're really popular in Japan. You put a ball up at the top. You usually flick the ball up at the top. And then that ball is going to bounce down. And if it eventually goes where you want it to go, if it eventually goes into a little cup down here at the bottom, then you might get more of those balls back. But in that case, it wouldn't. So imagine a game of pachinko where we had one ball like this that's bouncing its way down. And then we had, instead of another ball, we had like 8 balls that are attached together. So something like that. So what would happen to them if we dropped them in a pachinko machine? Does it make sense that they're sometimes going to go slower, they're going to wrap around, it's going to take them way longer to get down to the bottom. And so the single pachinko ball would already be at the bottom where this big chain of pachinko balls isn't even made it very far. And so how does that apply in DNA? Well, think of DNA instead of pachinko. So basically, if we were to take a small fragment of DNA, it's going to work its way farther down through that pachinko machine sooner than one of these big fragments. And again, we don't use pachinko machines, but we do use gel electrophoresis. And so how does this work? Basically, inside you have a gel. The gel is kind of the consistency of Jell-O. It's going to have little wells on one side where you insert the DNA. And what's pulling the DNA? Well, in a pachinko machine, it's going to be the gravity. But here you can see that there is a red cable. That means there's a positive charge on this end. So there's a positive electrode all the way across. There's going to be a negative electrode. You can see it right back here on this side. So basically, the DNA is going to migrate across that gel. DNA has a negative charge, so it's going to be pulled towards the positive end. And so if we look at this, this is kind of that, if you've ever heard of like a DNA fingerprint, what's going on? Well, basically the DNA is going to be pulled in this direction. We could say this, since it's been on for a while, that this fragment right here is going to be bigger than this fragment right here, because this small fragment has made its way farther from these original wells where it was up here. And lots of times you'll run a ladder as well. A ladder is a bunch of DNA with known distances or known quantities, and so you can read across and see how big is that, how many nucleotide pairs do we have. When we do this in class, we use a dye called ethidium bromide, and basically it'll dye the DNA. You can put it under black light and you can see where the DNA is. It's normally clear, you can't see it. Okay, what's the next thing we have to do? Remember, we have to, now that we've sorted it according to its length, now we have to make copies of it. And to do that, we use the polymerase chain reaction, or PCR. This was invented by Kary Mullis. He was riding his motorcycle one day and came up with this idea. And so to do that, we need a few extra things in our PCR machine. One thing we need is a primer. Primer is going to be a little section of DNA that'll grab onto the DNA. It'll allow that polymerase to drive down the DNA. Now, where's Taq polymerase from? Remember, it's that bacteria. Because in the PCR, we're going to heat this up really hot. What else do we need? We need nucleotides, so we need new letters. And so let's check this out. Basically, we put it in a PCR machine, which looks kind of like a photocopier. You put your DNA in the top, and then it's going to quickly heat it and cool it and heat it and cool it. And so as it heats this, it's going to unzip that DNA in the middle. It's going to break all those hydrogen bonds. What's the next thing that's added? Well, the next thing that will be added is going to be the primer. The primer is going to bond to the complementary sides of the DNA. Now, we create the primer because it's going to target a specific gene that we want. What happens next? Well, once the primer's in place, then Taq polymerase is going to grab on. Now, this looks familiar. If you know anything about DNA replication, how does that work? Well, we unzip our DNA. It's helicase that does that. We put a primer down, and then it's going to be DNA polymerase inside us that makes copies of our DNA. But in a PCR machine, it's Taq polymerase, and the reason why is Taq polymerase can withstand these really hot temperatures. Okay, watch carefully what happens next. Well, basically, as the Taq polymerase runs in either direction, it's going to add complementary letters to either side. And so basically, what do we have? We had one strand of DNA, now we have two. So the PCR machine will cycle. It'll again heat up the DNA, so it unzips those hydrogen bonds. We're going to add primer on either side. Taq polymerase is going to grab on, and as it races down, we're going to produce complementary sides. And again, heat it up, we're going to unzip the sides, the primer is added on, Taq polymerase on, and now we have, if you look at it, we've now got 8 strands of DNA. And so this just in a few minutes, we've gone from 1 to 2 to 4 to 8, and now we just get 16. You get that exponential growth. And so we can make a whole bunch of DNA very, very quickly. So we're almost to the end. What's the last thing in our analogy of the ransom note is really reading what those letters are. And to do that, we use a DNA sequencer. DNA sequencer, and sometimes it's hard to explain. Basically, if you were to Google the Sanger method, you'd find some videos that might help a little more. But basically, it's like a PCR machine. So if you look down here, we got the primer, we got the Taq polymerase, we got our letters, but then we have these 4 special letters. They're called dideoxynucleotides. And so they're just like adenine, cytosine, thymine, and guanine, but they have a specific color. And so what happens, you'll heat it up, primer will be added, Taq polymerase will be added, and then we're going to start adding those letters. And so if I were to go across, if there's a T here, there'd be an A here, and a T, and a G, and a C, and we'll say that's a T and a T. C, G, G, C, A, A. Okay, so usually it's just going to make copies of it. But occasionally, as it makes those copies, instead of putting a regular A in, it'll put one of these weird A's in there. And what that weird A is going to do is it's going to be dyed, it's going to have a specific color, and it's also going to stop the sequence. And so basically you can't add new letters. And so a lot of the time you'll get a regular run-of-the-mill copied DNA strand, but occasionally you'll get these fragments that just have an A at the front, and they have a specific color, which is going to be green. Maybe we run it again the next time, and the A seems normal, but the next one is a T that's weird. And so that's going to stop it at that point, but it's going to give it a color of red. Or maybe another time we're going to get an A and a T and a G, but then we're going to put a weird C on there, and that's going to stop it. And the C is going to have this blue color. And so basically what you can do is then run gel electrophoresis. All these fragments are going to run through a gel electrophoresis machine, or running a gel. And basically what they're going to do is they're going to separate according to their fragment length. Well, what's the smallest fragment which went the farthest? That's going to be this little fragment of A with the weird A at the beginning. And so what does that tell us? Well, it tells us that the first letter is going to be an A. And so a computer can go through and read all these colors, and it can read the sequence of the DNA. It can figure out the letters in our DNA. And so the Human Genome Project, that was the job of that, to sequence all of the DNA in a human. These are the DNA sequencing machines, but what I want you to remember is that we've sequenced all of the DNA in a typical human, and as you grow up, they'll be able to sequence all the DNA specifically inside you. But at this point, we're kind of right here. We've got the DNA, we know the order that it's in, but we don't know what proteins those make or how those proteins interact. And so the next project after the Human Genome Project is the Human Proteome Project. And so that's a lot, I know, but I hope that's helpful.